Welcome back. We're doing viruses today, Lab 12. This is on page 90 in your lab manual. Plan for today, talking about viruses, are they dead or alive? Infections, specifically the flu virus, pandemic versus epidemic, antigenic drift versus antigenic shift, vaccines, and then how does this all connect to COVID-19? And then talking about your homework. Hopefully to boost your mood, get you in an Easter mood. Here's my dog with some flowers. Had a little photo shoot with her every time I take her on a walk. So enjoy these. Daisy in her natural habitat. So viruses, are they dead or alive? Let's talk about living characteristics. What makes something that's living, living? So we have biotic and abiotic. Biotic means living and abiotic means non-living. So some characteristics would be genetic material in proteins like DNA or RNA, taking in nutrients, giving off waste, respiring, going through cellular respiration, reproducing, and then evolving over time. I say over time because as an evolutionary biologist, evolution doesn't take place very quickly. It's a slow process. So let's watch this video real quick about viruses. It's a rap that you will never get out of your head, so you'll never forget it.
I'm a virus, an infectious particle, an anathuga the genuine article, Ebola, chickenpox, West Nile, influenza, yellow fever, AIDS, herpes, SARS are coming right after. I'm not a cell, not an independent organism, don't even have my own metabolism. I only reproduce myself by taking over cells, then I bust them apart, no wonder you don't feel well. The structure is simple, mostly proteins and genes, packaged as a sub-micron killing machine. My capsule's made of capsules and self-assemble. Whenever cells see me, they start to tremble inside my capsid is where my genes reside. I got DNA or RNA on the inside, and the rest of me is various proteins. Whose role is assisting in assault cells and taking control.
I'm a virus, controls your cells around with it. I'm a virus, your cells is the life of me.
Yeah, so you'll never forget that. He's very funny and he has a lot of great science videos actually, so something to keep in mind if you need help with something. So, knowing what we know from living characteristics and seeing the video, here we can talk about what characteristics viruses share with living organisms. So green is things they do share, red is things they don't, and yellow is kind of an iffy level. So they do have genetic material and proteins, like he said. They have DNA or RNA in a capsid, in a protein shell. We can come in different shapes, like this is more helical, this is polyhedral, so kind of like a different kind of shapes. There's a wide variety of different shapes, and some of these can even have an envelope around them, lots of different things. They do not take in nutrients, they do not give off waste, they do not respire. He said they don't have metabolism. They can reproduce, but only with a host, like he said. So they do kind of share that characteristic with living organisms, but not really, because they can't do it on their own. They do evolve over time, which is why we see outbreaks like COVID, different strains coming to light. You've heard it said the novel coronavirus, so coming to light. So is it living or dead? We generally would say it's dead, because it's really just taking characteristics that living organisms have and manipulating them to get there, to attack and to take over and divide. So they do have the genetic material and proteins, but they're really not living. So a virus can enter the body in many ways. It can be breathed in, eaten, or acquired when bodily fluids are exchanged. But sometimes for each virus, only some of these pathways will result in an infection, and we call that tropism, basically the way they infect to cause an infection. And so we can have the virus enter the cell, it multiplies, and here it spreads out, goes out, and then each one can go and infect another cell, and it just repeats and repeats. Or it can inject genetic material into the cell. So you can see this picture of a microscopic image. Here we have these little tiny viruses, these capsids here. The DNA or the RNA is in here, each of these, and it injects it into this cell. This yellow part is the cell, so it goes in and injects the DNA. So the virus can either go in the cell itself, or it can inject its genetic material into the cell. And what it's doing there is it's taking over the cell's mechanism to make more and more viruses to attack and control. So an example is the coronavirus, actually. This is the mechanism that it undergoes. So we start over here. So we have the coronavirus over here, SARS-CoV virus, that's a specific name. And it has RNA inside it, so that's it. It's the genetic material. And then here, each of these spikes are different types of proteins that go around it. What happens is it binds to a receptor on the cell, so you could say in our bodies, and it goes in. Basically think of the receptor as a lock and key mechanism, and so it tricks our cells into thinking, oh, it's fine, it can come in, it's all good. So it comes in, it releases its RNA into our cells. Our cells, it basically tiptoes in. You can think of it as kind of it's standing around the corner and then it sneaks in, and it uses the cell's mechanisms to copy the RNA, which can do multiple things. It makes a lot of copies of the RNA, so we have tons and tons and tons of copies. And it also takes the RNA, which makes proteins, and it makes all the proteins that are needed to make the outside shells, those receptors we talked about. Once we have a ton of shells and we have a ton of those RNA copies, they assemble them all together, and then they send them out of the cell and attack. So if you think about it, you have one virus come and bind. You can see we already made 4 copies, if not more, and we sent out 4 more. And so if you attack each cell, you're multiplying and multiplying and multiplying over and over and over again, and that's why it moves so quickly. Viruses move very, very quickly. So let's talk about some more tropism, some different outcomes. This is on page 91. Specifically, we are talking about 4, and I have some 4 examples for those. So the first one is cell death. Basically, what's happening is the virus takes over the cell and takes over the machinery that controls the cell and shuts it down. And a good example of this is Ebola.
Blood clots begin to form. Internal organs begin to fail. And in a matter of days, the body hemorrhages and dies. Terrors, all caused by a nightmare, come to life. Ebola is a rare but extremely dangerous disease. It's classified as one of the most lethal diseases on the planet, with a fatality rate of up to 90%. Ebola is caused by 6 species of virus, with 4 known to cause sickness in humans, and each is named after the locations of their outbreaks. The first known species was Zaire Ebola virus, discovered in 1976 near Zaire's Ebola River, a river fated to become the namesake of all Ebola viruses. Like all viruses, Ebola viruses infect and feed off of their hosts, starting on a microscopic level. The viruses, which are string-like structures, contain genetic information in the form of ribonucleic acid, or RNA. Encasing the RNA are layers of proteins.
You can see here, this is our RNA is deep, deep inside, and then it has tons and tons of layers of proteins. So this is another example of a shape. Instead of being circular or polyhedral like we talked about, this is more of a string-like shape.
Proteins on the outermost layer are highly versatile, capable of changing shape and binding to different types of cells within a host. When binding occurs, the Ebola virus fuses with a host cell, allowing the virus's RNA to infiltrate the cell.
This is, again, another example of how it injects it into, it doesn't enter the cell itself, it injects it in, and then it makes multiple copies inside, and then they break out and infect other cells.
And replicate the virus from within. This infection quickly spreads to countless cells throughout the body, resulting in some of the most terrifying symptoms known to man. Within 8 to 10 days, those infected by the Ebola virus may experience symptoms drastically ranging in severity, from fevers to diarrhea and vomiting, to internal and external bleeding, often from the eyes. While outbreaks have been caused by multiple species of Ebola virus, the strain with the highest fatality rate was the first to be discovered, Zaire Ebola virus. In 2014, it caused the most severe outbreak in history, sweeping through Western Africa and resulting in over 11,000 deaths, more than all earlier Ebola outbreaks put together. At this point, no cure for Ebola exists. Plus, the communities most likely to be affected do not have sufficient access to health care. In the meantime, international efforts are being made to develop and test vaccines that could potentially stop the disease once and for all.
So you may have remembered the Ebola outbreak back in 2014. You may remember the Ebola outbreak in 2014. You may remember how horrible it was. So many people died very rapidly because basically the virus infects and destroys your cells, takes over and shuts it down. And you spread it very easily, and the traditional culture of the area where it was happening is they bury their own dead and they touch them, and so it's spread with bodily fluids, and so it can get spread very easily. So that's cell death, and this is going to be a little bit of a downer lecture, but just bear with me. It's very interesting. Another outcome is transformation. Basically, the virus activates cell proliferation, basically, which leads to increased and uncontrolled cell growth. So when we talk about cancer, basically what's happening is your cell has built-in mechanisms that control how fast and how quickly cells divide. And with cancer, that mechanism is lost, and so you have this uncontrolled cell growth. And one example of this is human papillomavirus, or HPV, which is a virus that is spreading, can cause genital warts and can cause a lot of different types of cancers. And so they have developed a vaccine for it called Gardasil, which is great. You can get it now. They used to only let you get it until you're 26, but now they let you get it until you're 45. Basically, it's a 3-step shot. You get 3 shots over 6 months, and it protects you from a lot of different things. 2 of the strains that it protects you against cause 80% of cervical cancer. It protects against genital warts, 90% of the genital warts, and then also against different strains that cause different cancers in different parts of your body. So it's it's a great vaccine. It's a great thing. So that is an example of transformation. Basically, the virus makes cells divide more and more and more and cause cancer. Another type is called latent infection. Latent infection, the host cell has a dormant virus that is in there for a long time, and it's activated later when something triggers it. So you can think about it like if you have had chickenpox as a kid, unless you've gotten the vaccine. If you had chickenpox as a kid, you won't get chickenpox probably ever again, and you won't get it for a long time. But then people who are older in their 60s and 70s can have the virus come back, triggered by something, and they get what's called shingles. So basically, the virus is in there for a long time, and then it becomes reactivated. Another example, outcome is chronic slow infection. So viruses that only cause infection after years and years. And so one of those is HIV. So we'll watch another video on that.
About 37 million people around the world are currently living with AIDS, making the disease one of the worst pandemics in modern history. AIDS, or acquired immunodeficiency syndrome, is a disease in which the human immune system is severely impaired. It's caused by the human immunodeficiency virus, or HIV, and is the last stage of the virus's infection.
I just want to touch on that you get HIV, the virus, and that leads to AIDS. AIDS is the last stage in having the virus.
HIV attacks by entering the bloodstream and attaches to helper T or CD4 cells.
Those are cells that are in your immune system.
A type of white blood cell, CD4 cells are responsible for fighting infections. First, HIV attaches itself to a CD4 cell and fuses with it. Then, HIV releases its genetic material into the CD4 cell, which combines with the cell's DNA. This allows the virus to produce more HIV proteins inside the cell.
We call this a retrovirus, retro meaning backwards or before. Basically, the idea is it uses our own DNA and puts itself in there so it can be copied more and more and very sneakily. We don't notice it for a while.
The proteins are then packaged and released to attack other CD4 cells. Without HIV medicines, an individual's infection may spread and within 10 to 12 years advance to become AIDS. This decreased immunity makes patients with AIDS vulnerable to a number of diseases, including infections, dementia, and cancer. Scientists believe that HIV originated in West Central Africa, where a similar virus was found in chimpanzees and gorillas. Around the turn of the 20th century, the virus was transmitted to humans due to the handling of chimpanzee meat. The virus then spread from the African continent during the mid-1960s through a single carrier, eventually making its way around the world. In 1982, scientists labeled the illness as AIDS, and the following year identified the virus as HIV. Ever since, about 77.3 million people around the world have contracted HIV, with the majority of affected individuals living in countries with very few resources. Scientists are working to one day develop a cure for AIDS. In the meantime, advancements in biotechnology, plus increased awareness, prevention, and access to healthcare, are saving many lives and may hold the key to finally exterminating the disease.
So, as you can see, HIV basically infects, and it doesn't cause an infection for, it causes problems for a long time, but it doesn't lead to as many problems until later on. So we call it a chronic slow infection. You're causing lots and lots of problems, lots and lots of time, you're causing infections over time. So those are some examples of different outcomes of infections, different mechanisms. Let's talk about the flu virus.
The flu is a highly contagious respiratory illness. It turns up year after year with devastating consequences, all caused by a most elusive virus. The influenza or flu virus is a recurring nightmare. It causes more than 36,000 deaths in the US each year and was responsible for some of history's deadliest pandemics. Like other viruses, the flu virus is a parasite. The viral agent itself, called a virion, is made of a ribonucleic acid, or RNA, surrounded by proteins.
So again, an RNA, genetic information surrounded by proteins.
The flu virus uses two proteins to attack its host, hemagglutinin and neuraminidase, or the HA and NA proteins. The HA protein attaches the virus to a cell and lets it in. Once the virus is inside, it multiplies. Then, the NA protein cuts this viral swarm loose, sending it off to infect more cells. This triggers the immune system to bombard the swarm and to destroy infected tissue throughout the respiratory system. Unfortunately, this response can also inadvertently lead to death through organ failure or secondary infections like bacterial pneumonia. The flu virus has been nearly impossible to eradicate, largely due to its uncanny ability to mutate. Since just 2004, more than 5,000 different strains of the influenza virus have been sequenced. Those that affect humans are categorized as types A, B, or C, with type A strains being the most capable of unleashing a pandemic. Influenza A viruses mutate more rapidly, allowing them to adapt to new hosts and even cross species. Avian flu and swine flu, for instance, are two strains of type A viruses that, through mutations, can be transmitted from birds and pigs to humans. One of the deadliest flu pandemics was caused by one such virus. Between 1918 and 1919, the Spanish flu infected a third of the global population and killed up to 50 million people worldwide. Called H1N1, this particular strain of virus likely came from birds. The threat of another influenza pandemic remains. In the meantime, scientists are constantly monitoring the flu virus and developing seasonal vaccines to create our best line of defense.
So yes, you may or may not get the flu shot every year. It's very serious, and I know even in light of COVID-19, it doesn't seem that important, but it is something important to still be thinking about. So like they said in the video, it uses different things to fool the cell into taking it in and replicating itself. It has these proteins on the side called hemagglutinin, or HA, that bind to the cell receptor on the cells called sialic acid, binds to those, and that lets the flu virus into each of the cells. So you can see those are these little spikies here. Lets it in, it replicates everything inside, makes new flu viruses, and the way it escapes is it uses neuraminidase, Na. It breaks those molecules, the sialic acid molecules, and it lets it escape out of the cell. A good way to remember this is, hey, come on in, nah, I'm gone. Hey, hemagglutinin, it lets it in. Neuraminidase, nah, I'm gone, it leaves. So here is a more zoomed in picture. So you can see here's the virus. We have neuraminidase and hemagglutinin. It binds to these sialic acid receptors on a normal cell, which lets it in. The virus multiplies, and then as a way to escape, the neuraminidase breaks that bond, and it escapes. So again, connecting this back to COVID, this is the picture I showed you earlier, similar thing. It may not be neuraminidase and hemagglutinin, but a good example of how it uses a receptor, it binds, goes into the cell, and then here it doesn't use a receptor to escape per se, but it does have that receptor to enter the cell, which they call the ACE2 receptor. So they mentioned in the video the different type of flu viruses, and there's A, B, and C. A is in most things we see around the world: ducks, pigs, horses, poultry, humans, animals, all kinds of things. Whereas B and C are only in humans, and they've never caused a pandemic. A is most likely to cause a problem and be spread around the world because it's in so many different things. Now, we've been mentioning pandemics over and over again, and you've probably been hearing it in the news all the time, but what does that exactly mean? Well, pandemic is a disease that's spread around the world. Pan means earth, so it's spread all over the world. An epidemic is an infection, a disease that causes an infection in many people in one area for a short time. So like the Ebola epidemic was only really in that area of Africa and Zaire. It could have spread. We had people come to America with it, but we were able to stop that very quickly. And so it's just an epidemic. Pandemic, all around the world. Epidemic, in one area for a short time. So you could say pandemic is all around the world. Epidemic is like if everyone in Cincinnati got sick all at once. So is one worse than the other? Not necessarily. You can have a pandemic all over the world, but only like one person in each country is sick, or you can have an epidemic where like the largest city in the world, every single person is sick. Doesn't necessarily mean one is worse than the other. It's just a way of classifying. Coronavirus, pandemic or epidemic? It started as an epidemic, mainly in China, and then as it spread all over the world, they classified it as a pandemic. In general, I think they classify it as a pandemic if it's in every continent, minus Antarctica. So now let's talk about antigenic drift and shift. This is how we see things that change and become new, novel types of infections and viruses, which is what we're seeing more and more. So let's talk antigenic drift first. Antigenic drift is when we see small mutations. Usually it's a mistake when the DNA is being copied in the cell. And so we're seeing just a slight change. It occurs very slowly, and it's not as dangerous because it moves very slowly. And if it goes through a population, the people's immune systems are able to have time to react and can remember it very easily. This picture shows a good example. Each of these is a receptor, like we've talked about. Starts here, small mutation. Instead of a circle, now it's a semicircle. But we're able to adapt very quickly. A good way to remember antigenic drift, DNA. Mistake in the DNA. Now, antigenic shift is when viruses infect one cell and they swap genes to become a new or novel virus. So here in this picture, you can see we have 3 different types of receptors, purple, green, and yellow, and they swap genes to become this new strain which has all 3 types. This picture too is talking more about how we got the influenza A, H1N1, the Spanish flu that they mentioned before, or even if you were aware of the swine flu back in, I think it was like 2009 or 10. I remember my high school play was, we all had to wear masks in order for it to go on. But here we have red for pigs, blue for chicken, purple for humans, and this other green for another type of pig, and basically they all swap genes here, and then we have this unique one. We're not as ready for it because we don't have memory cells to remember it. We haven't been infected with different types like this, so we can't react to it. If, let's say in this picture, we are only used to the green ones, and we've never encountered purple and yellow, when we get this going into us, we're going to be like, what? We don't know how to adapt. We think this is maybe similar to what happened in coronavirus. They think it originated in bats, which then probably led to pigs being infected, which probably happened at some meat market, maybe not pigs, but some kind of meat. Something was eating food around where bats poop. Something ate it, probably a pig, and then we ate it as humans, and that led to an infection. So we were not able to adapt very quickly, and it moves very fast. Before we go back to COVID, I want to talk about vaccines real quick. So if you go to page, let's see, 93, 94. So the best way to fight a virus is to not get it, and usually that means a vaccine. If you think about vaccines, you've heard of flu, measles, mumps, rubella, chickenpox, yellow fever, Hep A, Hep B, HPV. These are all, which the Gardasil vaccine, these are all things that we fight against with vaccines. There are fewer infections against bacterial diseases, but we do have a lot for viruses. So a vaccine is usually an inactivated or attenuated, which means a weakened virus or bacterium. Usually it's just a few proteins from the pathogen, or some kind of version of that. It can be either from the actual virus, or we make a synthetic one. And then basically you get the vaccine, and that teaches your immune system cells to recognize the virus and then fight them off if you have a real live virus in you. So is it harder or easier to make vaccines with viruses? It's harder because of antigenic shift, because the virus can evolve in unvaccinated people. So if you get a flu shot, you get a flu shot every year, and you may think, oh, well, I only got chickenpox once. Why do I have to get the flu shot multiple times? That's because of antigenic shift. Because people don't get the flu vaccine, it can mutate and evolve. We'll call it, it'll mutate and accumulate genes from other places and change in other individuals. So then we have to keep getting vaccinated against it. And actually each flu vaccine is for a specific strain or combination of strains that we're thinking is going to be the worst one that year. It may not protect you against every single strain because, like they mentioned in that one video, there's tons. They said like 500 to 5,000 different strains. That's why it's so important to get a flu shot. Even if you're like, I've never gotten the flu, it's fine. It's important to remember that the virus can actually change in you, the antigenic shift, and so then even if your grandma is vaccinated, she's not ready and prepared for the strain that you have, so she could still get it. So very important to get a flu shot, even if you don't think it matters. It's important to protect people in our society, especially older people and newborn babies and people with immunodeficiencies. Now let's talk a little about COVID, some application. If you've seen the movie Contagion, it's amazing. And if you haven't, I highly recommend. It may not be the greatest time to watch it because it's kind of scary and very applicable to what we're seeing today, but I usually show this in my class, and I wanted to show you the trailer and then the ending, and it's not going to ruin anything. It just talks about how the virus itself was spread, and it relates very amazingly to what we're seeing today. So let's watch the trailer first.
It was a groundbreaking ceremony for a new factory. Did she mention seeing anyone who was sick? Anyone on a plane at the airport?
No, she said she was jet lagged.
The average person touches their face 3 to 5 times every waking minute. In between, we're touching doorknobs, water fountains, and each other.
Beth. No, go up to your room, honey.
So we have a virus with no treatment protocol and no vaccine at this time.
You had a seizure this morning, Beth.
She had a history of seizures and allergies.
As of last night, there were 32 cases.
Unfortunately, she did die.
Right. So can I go talk to her?
Mr. Amoff, your wife is dead.
What are you talking about? What happened to her? What happened to her?
Is there any way someone could weaponize the bird flu? Is that what we're looking at?
Someone doesn't have to weaponize the bird flu. The birds are doing that.
Watch this. It's transmission, so we just need to know which direction.
On day 1, there were 2 people, and then 4, and then 16. In 3 months, it's 1 billion. That's where we're headed.
They're calling out the National Guard. They're moving the president underground. People will panic.
Get away!
It will tip over.
The truth is being kept from the world.
Cook your samples, destroy everything.
Hello. I need you to get me the names of everyone who serviced this room. It's an emergency.
You can't panic now.
I know.
I'm going to get you home. I got people too, Dr. Cheever. We all do.
Don't talk to anyone. Don't touch anyone. Stay away from other people.
Get back in your car. We're not sick.
It's figuring us out faster than we're figuring it out. It's mutated.
So, like I said, highly recommend. Such a good movie. As a scientist, I love it, but also look at the cast. Amazing cast. And man, watching that again, it really hits home how applicable it is to right now. It's mutating faster than us. Why weaponize the bird flu? The birds are doing that. Exactly. It's just, chills. It's amazing. Let's watch the ending. And again, it doesn't tell you what happens. It just talks about how the virus itself was spread. And we'll talk about how that applies. Just to preface, this happens at the end of the movie. After everything happens, it just shows us some background, flashing back to what happened before the contagion happened. So, what is this? What does that mean? What are we seeing? So basically, bats ate something. Bats had the virus. They dropped some food down. The pig ate it. Then the person touched the pig that had it. And then the person shook the hands with Gwyneth Paltrow. And that was day 1. That's where we saw the first outbreak. And so what does this mean? This is basically showing antigenic shift, right? It's changing, swapping genes in different organisms. And so then we are not prepared to handle that. And you can see how quickly it moves. And this is similarly to what they think is happening in COVID-19. Coronaviruses are a type of virus that originate in a lot of different places, but they do think in bats, and they do think it was spread to some animal in a meat market in China, which then was eaten and was spread to humans. Kind of similar to how we saw in the HIV outbreak with the chimp meat, kind of a similar idea there. So this video I won't show you right now is a great background on COVID-19, what it means, where we've gone. This is an older video actually from last month, so it's not totally up to date. But this is a nice graphic from the state of Ohio's COVID website. You can Google, it's coronavirus.oh.edu or different websites per state, but it shows you basically which counties have it and how many cases, and then it shows you the total cases per day and the total deaths. So this is from April 8th today, and as you can see, we are in Ohio starting to plateau, which that is the good news. From yesterday to today, we've only gone up about 4 cases. Now there is delays, of course, but we are starting to plateau, which is great. We're starting to flatten the curve, which means we are spreading it less, and our medical system can handle the number of cases better. So I would encourage you, even if you're not from Ohio, or if you are from Ohio, to please stay inside. Please keep doing what you're doing and washing your hands, not exposing yourself. If you feel like you have been exposed, staying home for 14 days and not spreading it. The more we can keep it down, the sooner we'll be able to come back and have more of a normal life, go back to school. I know it's hard to live like this and to accommodate it like this, but the longer you fight it, the longer you're going to be inside and not having a normal life. Some states are predicting that they won't even peak or plateau until like June or July, and at that point, what's the fall going to look like? It's going to be crazy. So just please be careful and be wise out there. So that's Lab 12. Let's talk about homework for next week. We're wrapping up this class. We don't have very much homework left. So make sure you're getting all your points in. I just want to also reiterate that all assignments are individual. So make sure you're doing them on your own. So for next week, you have a graph. I told you last week that I touched some MSA plates with my fingertips. My husband did it and my dog. And you're going to make a graph of the results. And I'm going to talk about that in a minute. That'll be due on Canvas. And it'll be due either the 23rd or the 24th. Check with your professor about when it is due. The rubric is on page 97 in the lab manual and on Canvas for my class. We are going to have another Popular Press article. I found a Popular Press article about coronavirus. It's a little outdated now, outdated meaning a few weeks old. And you're going to answer the 6 questions that are on 98. It's just like the normal Pop Press articles we do. You should just know that it is a little longer than we normally encounter because it is such a new and interesting topic. So it is a little longer, but trust me, you'll find all the answers in there. You're going to do the Lab 12 lab report, the worksheet similar to what we've been doing. This one will be about viruses. Again, do either the 23rd or the 24th. Check with your Canvas specifically. Open note. Email us if you have questions about it, but yeah, open note, 20 points. And then something we're doing is your next graph, and you're going to write your own Popular Press article about it, is I swabbed different things in my house. Normally what we do when we're on campus is we give you swabs and we say, go swab whatever you want. People do phones, they swab Gallagher keyboards, bathrooms, things like that. And then they count the number of bacteria on the plates and write a Popular Press article about it. Now, because you can't do that on your own, I did it for you, and we wanted you to still have a little shred of choice. So you can basically pick if you want to do phones, keys, laptops, or doorknobs. So I swabbed the phones in my house, our keys, our laptops, and our doorknobs. So be thinking about what you would potentially like to write a Popular Press article about, something that you're interested in, and you'll pick one of these. Now, you're going to sign up in different ways. Dr. Matre has a sign-up sheet for her class, and I'm using a Google Sheets sign-up that is on the last slide of this PowerPoint. For you to sign up, you need to sign up by the 24th, just so I know what you're doing. All of the information about this graph I will explain in the next video, but just sign up by the 24th. So again, be thinking about what you want to do. Something interesting about doorknobs, it may seem boring to you, but there's studies that show that doorknobs that are metal don't grow a lot of bacteria on them actually, so should be interesting to see. Okay, let's talk more about the graph. So for the graph, you have the choice of doing a graph or a table. Let's talk first about the graph. You're going to graph the data. This is due, again, the 23rd or 24th. You're going to use the photos on Canvas. I'm going to show you at the end of this where they are. You're using pictures I took of plates on Canvas, and it's in the folder called the fingertip photos. And you're going to see RC, TC, and Daisy. That's my husband, that's me, and Daisy, my dog. Basically what you're going to do is you're count up the colonies in each circle of the different kinds, and you're going to distinguish them as these three different ones, Staphylococcus aureus, epidermidis, and Micrococcus luteus, like you did with the worksheet. But you're going to count how many you find on each one. You're going to count how many of each you find for Ryan, for me, and for my dog. You're going to add them up, and then you're going to average them. So like, for example, you could say, oh, Ryan had 140 Staphylococcus epidermidis, I had 20, and Daisy had 1. You add them up, that's 161. You divide by 3 for each of us, and there's about 53.3 Staphylococcus epidermis on average. Then you'll make a graph. Now, for the graph, what I would recommend, I made little baby graphs here. Do a bar graph or a pie chart. For the bar graph, what I want you to do is you'll have each of us, so Ryan, me, and Daisy, and then you'll have a bar that's the average so we can see how it compares. So here I have Staphylococcus epidermidis, Staphylococcus aureus, and Micrococcus luteus. These are all made up numbers, by the way. So you can see, oh, you know, Ryan had more than me and Daisy, but the average is about 4.5. Again, made up. Or you can do a pie chart. For that, you do not need to have each individual one listed. You just need to have the average. So here I have Staph epidermidis, Staph aureus, and Micrococcus luteus. I like to have the percent or the number in it just so I can see. Those are the graphs. Or you can do a table. Again, very similar, but what I like to do, and what I would recommend for you, is we have Staph aureus, epidermidis, and Micrococcus luteus, and I would like to mention that they should all be in italics because they are bacterial names, and that is my fault for not italicizing this. And what you'll do for the table, you have all three here, is you'll have Ryan, me, and Daisy, and then the average. You'll say, oh, Ryan had 20, Taylor had 10, and Daisy had 1. And then the average of all three. You'll do that for your table. Again, for the graph and table, you will need a caption of some sort. The only difference, and this is again on the rubric on 97, the graph, where does your figure caption go? Figure final goes underneath it. For a table, your table caption goes on the top, table top. So your figure caption needs to be up here. It's basically the same thing, but it's just at the top. Okay, now I'm going to show you where to find the file for the fingertip data. So this is your student view under Canvas for my class. It's under Files, and you'll go to MSA Fingertip. Ooh, I can't show you right now though. So let me leave the student view. You'll be able to see these as soon as possible. So here are the plates. I've included a little comment here so that you know things to note, the color change, and this black dot is mold, so you wouldn't count that as bacteria. These are very nice quality photos, if I do say so myself, so you can download them and zoom in as much as you want. Basically what you'll do is you'll count each type, like I said. I've included 2 pictures for Ryan's data because, so here it is. It's a little hard to see because they're starting to grow into each other. So here I kind of held it up to, there's my dog, but so you can see the color change. And then here's my fingertips. So if you have any questions, please let us know. Looking forward to seeing your graphs and your assignments. Please get in your assignments. This is really your only chance. You're starting to lose chances to get points before the end of the semester. So let us know if you have questions, and have a good week. Stay safe.